Hi and Assalamualaikum. My name is Nur Safwana binti Muhammad Safi'i, last year student of degree in biology from class AS2016A. In this video, I'm about to present my final year project with the title Diversity of Epiphytic Terrestrial Algae on tree trunks of Sri Gading Educational Forest, Pahang, Peninsular, Malaysia. This video consists of research background and objective, problem statement, methodology, results and discussions, and lastly conclusions. Algae are chlorophyll-containing organisms that mostly can be found in aquatic habitats such as marine and freshwater bodies. They also can be found in terrestrial habitats. Terrestrial microalgae belong to three different evolutionary lineages, which is a chlorophyta, often called green algae, stratophyta, and diatoms are known as Basilaриophyceae. Currently, there are 800 species known in the world. To distinguish one species from another, their morphology can be referred. They consist of three distinct habits, which are first, individual cells such as corolla, Chrolocrecium, Chrolorcoccum, and Trobesia. Next is sarcinoid behavior, which is packet-like colonies, and lastly, unicellular filaments. Are there branches or not such as Princia, Rosivigila, and Trotophilia morphology? The objectives of these studies are listed as follows. First objective is to identify species, and second, to analyze the compositions and abundance of terrestrial algae in Sri Gading Educational Forest. The association between algae and some other elements in terrestrial ecosystem has rarely been studied, leading to a fundamental lack of knowledge concerning their presence and terrestrial ecosystem. Besides, insufficient approaches and data interpretations regarding algae as biological marker of PTEs is one of the factors for the dearth of information on algae. Hence, several difficulties may arise throughout the research study, such as inability to identify the algae species due to their comparable traits. Two methods were used for completing the study. For algae collection, random sampling was used. Total 10 trees were randomly selected with presence of microalgae on the tree trunks. Later, the specimens were analyzed by their morphology and identified the species using World's Algae Listing Database. Thus, PAST software, or known as PAST software, was used to analyze the diversity value of the terrestrial algae. To achieve second objective, the diversity and abundance of terrestrial algae, two methods were used, which is Simpson Diversity Index and Shannon-Wiener Diversity Index. As for the result, a total 11 species were identified that come from a phylum of Chlorophyta with 2 classes, 2 families, 2 orders, and 4 genera. The first species found is Rosenvingilla radicans from 3 samples of specimen. These species were found at the forest entrance with green-colored appearance on the tree trunks. Second species found is Trentofolia diffracta, collected at inside and outside of forests and near the schools of wood tech sites. These algae form a dense orange coating on tree trunks. Third species is Trentofolia arborum, dominant species found inside the Sri Gading forest with green-orange light color. And lastly, species discovered is Trentofolia abitina, the most dominant species outside the Sri Gading forest with golden-orange mats of thallus seen on the tree bark. The ultimate factors for the abundance of alga species is associated with bark roughness, pH, and light intensity. Study shows that there are significant positive correlations between abundance of cyanobacteria and bark roughness. Also, open non-shaded areas such as forests can increase diversity and abundance of cyanobacteria and Trentopholiaris. However, there are also several factors that can affect algal growth and composition, such as excessively pollutant environment. The value of Shannon-Wiener and Simpson Diversity Index is 0.99 and 0.73 respectively. The more excellent value or close to 1 indicates that the species is diverse in that location. Even though both values are quite high, this result cannot be simply interpreted as high diversity of terrestrial algae, as the sampling intensity was quite low with short period of sampling activities. In conclusions, 11 individual algae have been identified with 4 different species under one phylum, which is Chlorophyta. They are Rosenvilla radicans, Trentophila diffracta, Trentofolia arborum, and Trentophila abitina. The diversity and abundance of these species was quite high due to the humidity, light intensity, pH, and structures of the bark tree. However, those indices not truly reflects the diversity of terrestrial algae in Sri Gading educational forest, since there are more species need to be discovered. This is end of my presentations. Thank you and Assalamualaikum.